Hello everyone, welcome back to Illum Biology class number 36, and today we are continuing with our topic isomerism in monosaccharides. This is the fourth part of the isomerism. I would request you to first watch the last 3 classes on isomerism in monosaccharides so that you can understand this lecture very well. So in this lecture we are going to discuss about optical isomers. Now first of all, let's discuss what is plane polarized light. Now in a normal light ray, we all know that a light is an electromagnetic wave, and in electromagnetic wave in light the oscillations are in the multiple planes as seen in this diagram. Now see, in this case you can very well see different planes in which oscillations are taking place. So in a randomly oriented light there are multiple planes in which oscillations take place. But when we pass this randomly oriented light through a polarizing filter, now what is a polarizing filter? What this filter will do, this filter will filter out all the planes of the light except one. That means after passing through this polarizing filter, the light will oscillate only a single plane, and that's when you call such light as a plane polarized light. That means what is a plane polarized light? A light in which oscillations take place only in a single plane, and normally the oscillations, the vibrations are in multiple planes. See, this polarizing filter has filtered out rest of the planes of oscillations, so there is only a single plane remaining wherein the oscillations take place in a plane polarized light. And now look at this carefully. What I have done, I have taken a solution of a compound in a beaker, and now I am passing plane polarized light through it. Now after passing through this compound, if this light does not show any variation in the plane in which it was oscillating, then this type of compound is an optically inactive compound. On the contrary, an optically active compound will always deviate the plane of the light in which it is oscillating. As you can clearly see in this picture, this is an optically active compound, and when plane polarized light, that is PPL, when it passes through optically active compound, then the plane will deflect from the original plane by an angle, let's say alpha. Now how do you define that? When a beam of plane polarized light passes through a chiral compound, it interacts with the compound in such a way that the angle of oscillation will rotate. Then the plane in which the oscillations were there will rotate in case of an optically active compound. And an optically active compound always contain a chiral carbon. It is always a chiral compound. What is a chiral carbon? A chiral carbon is a carbon whose all valences are fulfilled by different kinds of groups or atoms. Then in this case this is called as a chiral carbon. So a compound with a chiral carbon is a chiral compound and is an optically active compound. That means it will rotate the plane of the plane polarized light. Now on the basis of the deflection, these compounds, these optically active compounds, are of two types: dextrorotatory compounds and levorotatory compounds. Now in the case of a dextrorotatory compound, as I told you, dextro, this word has been derived from dexter, and dexter means right. So if an optically active compound deviates the light towards the righter side or towards the clockwise direction, then such a compound is a dextrorotatory compound. You can clearly see that the plane polarized light has been rotated in a clockwise direction, in this direction, clockwise direction, or on the right side, then such compounds are dextrorotatory compounds. On the contrary, levorotatory compounds will rotate this light towards the left side or in the anti-clockwise direction. You can clearly see that the light has deviated in the anti-clockwise direction. So such compounds are called as levorotatory compounds. And how do we denote them? We denote them by either a small d or a plus sign. A small d or a plus sign represents a dextrorotatory compound, and a small l or negative sign represents a levorotatory compound. Now I'll make one thing very clear here. You must have heard about D form and L form of monosaccharides, which are two different kinds of enantiomers. See, this capital D and capital L isomerism is totally different from small d or small l type of isomerism, and these two are not related to each other. Let's see how. See, this is how a glucose is represented by a Fisher formula. Now, how do you write about this structure? See, this is very simple. We write it as a D plus glucose and L plus glucose. First of all, this D, this is capital D, so this is not dextrorotatory. A dextrorotatory is represented by small d. This D means that this is a D form enantiomer of glucose, and this is the L form enantiomer of glucose, and this isomerism, as I told you, has got no relation with the optical activity, that is small d or small. As we discussed in the earlier videos, now what is a D form of glucose? It is that form of glucose in which the OH group on the last chiral carbon, see, this is the last chiral carbon. If the OH group on the last chiral carbon is on the right side, then this is a D form of glucose, and if this OH group is on the left side, then this is the L form of glucose. That's why I have mentioned it as D glucose and L glucose. Now, in this positive sign means that this is dextrorotatory. It's the same again, positive sign, that means it is dextrorotatory. So glucose is a dextrorotatory sugar, remember this, and this is the reason why glucose is also called as dextrose. So remember, glucose is dextrorotatory, which has got two enantiomers, capital D and capital L, and it is always represented by this plus sign. On the contrary, fructose is always levorotatory. That's why fructose is represented by a negative sign. These are the two different structures of fructose. You can clearly see that fructose is a ketose sugar. That's why a ketonic group is there. Now, first of all, look at the last chiral carbon. This is the last chiral carbon. OH is on the right side. That's why we have mentioned it as D minus fructose. This D represents OH on the right side, and minus represents the levorotatory nature of the fructose. Similarly, in this case, OH is on the left side. That's why we have mentioned it as L form of fructose, and minus will always represent the levorotatory nature, and that's why fructose is also called as levulose. You must have heard these two words, dextrose and levulose. Dextrose is basically glucose and levulose is fructose. Now, what is a racemic mixture? Let's see, in this case what I have done, I have taken a solution in the beaker, and this solution contains 50% glucose, for example, and 50% fructose. Now we all know that glucose is dextrorotatory and fructose is levorotatory, and in this case we have taken equal concentrations of both of them. So the dextrorotatory activity of glucose in this case is being cancelled by the levorotatory nature of the fructose. That's why in this mixture there would be no deviation in the angle of plane polarized light. When I pass this plane polarized light through this solution, which has got equal concentration of a dextrorotatory and a levorotatory compound, it is not deviating because of the cancellation of the opposite activity by the opposite nature of both dextrorotatory and levorotatory compounds. So such mixtures, such solutions are called as a racemic mixture. If D and L isomers are present in equal concentration, it is known as a racemic mixture or a small d small l mixture. Small d small l is dextrorotatory and levorotatory, and whenever a plane polarized light is passed through this racemic mixture, there will not be any deviation in the plane of oscillation of that light, because dextrorotatory and levorotatory activities, they cancel out each other. Now the question that arises in our mind is, sir, what things will decide the angle by which this plane is deviated? Let's see this plane, let's say this plane has been deviated by an angle alpha. Now different compounds will have different value of alpha. So on what factors this alpha depends? So this alpha, the angle depends on the temperature of the solution, it depends on the wavelength of the plane polarized light used, the concentration of the chiral compound, and the path length. What is path length? It is that length through which light travels through that sample. In this case, this is the path length because this is the length of the sample through which the light will travel. So this alpha will also depend on this path length. So that's it. Thanks a lot for watching us. Hope you liked this video. Stay connected with us for more such interesting videos. Thanks a lot.